Hello everyone, and welcome to phyloseminar.org, a project supported by the Society of Systematic Biologists. For this session, we're hearing from three courageous individuals who are working to incorporate structural information into molecular evolution studies. The previous talk was from Jesse Bloom, in which he described his work doing deep mutational scanning experiments to develop per-site models of amino acid substitution for specific genes. Today we're going to hear from Claus Wilke, and the next talk is going to be from Richard Goldstein on May 25th. Let me remind everyone to use the Q&A app to ask questions, and note that Claus has explicitly said that he prefers to have some questions during his talk. As I mentioned, today we have a talk from Claus Wilke. Claus received his PhD in theoretical physics from the University of Bochum in Germany in 1999. He was then a postdoc in the Adami lab in Caltech from 2000 to 2005, where he did training in biological physics, evolutionary biology, and artificial life. This included co-authoring one of the most highly cited papers on the artificial life platform Avida. I highlight this because Claus's research program still maintains a strong theoretical component concerning fundamental questions about how evolution works, but also goes all the way to much more specific and practical questions concerning the biophysical constraints on protein evolution, which is closer to what he'll talk about today. Claus is a full professor at UT Austin, and in addition to leading a very productive research group, is also currently the chair of integrative biology. For that reason, we're especially thankful that you're participating today, Claus, and I'm looking forward to your talk.
Yeah, thanks a lot for the invitation. This is actually the first time that I'm giving a seminar over the web, so I'm a bit nervous, but I hope that it will work out. And that's also why I would welcome some questions along the way, because I normally like to look at my audience and see how they react, and I don't have an audience now, so this feels a bit unconventional, but I'll make my way through it. Okay, so what I'm interested in fundamentally is explaining this. And now the slide didn't advance. Okay, there. Okay, this is a protein alignment. And as you can see, it looks kind of messy and there's a lot of stuff going on. And if you look at this, then you might just say, well, is there any sort of pattern in there? But if you look more carefully, you find that there are actually a lot of patterns. For example, there are a couple of sites that are completely conserved, right? They're kind of hidden. They're not necessarily right next to each other. But if you look at any of these, like, for example, this one is all P's, this one is all G's, this one is all K's, and so on, right? And then some sites right next to them are mostly conserved, but not quite, right? So for example, I had a completely conserved site here, a completely conserved site here, and then right next to them are sites that are mostly, but not entirely conserved, right? I have a lot of N's, but then here it starts changing into Q's and H's, here P's all the ways, and then some S's, some N's, and so on. Over here, it looks more like some biochemical property conserved, some valines, leucines, isoleucines, so all biochemically somewhat similar amino acids that seem to be rapidly changing at this site. And then finally, there's sites that are quite variable, like somewhere here in between, you see there's a lot of variation along those sites. And so if you look at this, just at the sequences, you might say, well, I really don't know why this particular site is variable and this site over here is conserved. But if you take the same alignment and map it onto a structure, things are starting to look much cleaner. So this is the structure from which this alignment was taken. So the particular protein here is E. coli exonuclease 3. That's the particular structure I'm showing. And then the alignment was, of course, homologous exonucleases from actually from all over the tree of life. And so here I've color-coded the evolutionary rate. There's many ways that one could measure that, and I'm going to gloss over the details in this talk, but it's basically a measure at each side of how variable that side is. And so in this graph here, dark colors mean low variability, the site is mostly conserved, and light colors mean high variability, the site is mostly variable. And you can see immediately that there is this pattern that the outside is more variable and the inside is more conserved, right? So I've highlighted here the surface side, and this particular surface side is very variable. And then I've highlighted here an interior site that is quite conserved. And you can see, for example, here, how as you go along the loop, you actually have all the variable sites on the outside and all the conserved sites on the inside, right? And so in the structural context, this makes perfect sense that the more conserved sites seem to be all towards the inside, the more variable sites seem to be more towards the outside. But if you look at this in the linear alignment, it would just look like a random pattern, and it wouldn't really be clear why this side is conserved and this side right next to it might be quite variable. Now, there's an additional element here, which is this is an enzyme, and there's catalytic residues, and I have highlighted all the catalytic residues here in white. So the white does not mean highly variable. The white just means this is a different residue, a catalytic residue. And in fact, what you can see is that the catalytic residues are right where you have the most sequence conservation. So everything right around the catalytic residue is quite brown. So the conventional explanation would be that these sites need to be conserved because the enzyme needs to function, and therefore these sites and their neighboring sites are conserved. But then they're also in the interior of the protein. So now we don't quite know, are these sites conserved because they're next to the catalytic residue, or are they conserved because they're in the inside of the protein? And most of what I'm going to talk about for the next 40 minutes or so is going to be this question of how does the interior versus exterior conserve protein sequences, and how do catalytic residues conserve protein sequences. And then I have a third part at the end where I'm going to talk about protein-protein interfaces. Okay, so let's first start with part one. How does protein structure constrain site-specific evolution? So let's forget about function for a moment. Let's think only about structure. So this is a question that many students and postdocs in my lab have worked over the last couple of years. So I'm highlighting specifically Amir Shamoradi, Austin Meyer, and Alicia Jackson. And then for the last 2 or 3 years, this project has also been really pushed forward by what I call a collaborative competition with Julian Chavez. So in particular, the next few slides will mostly talk about papers that either Julian wrote or I wrote. Lately, we've also started writing papers together. It's actually really good to have another senior person in the field who has a slightly different perspective, but does very good work. So then you can push yourself forward, and I'll outline that in the next few slides. Okay, so it's been known for a long time that surface residues in proteins are variable and interior residues are more conserved. So that's not really anything new. But what has happened over the last maybe 6 to 7 years is that people have tried to really be more quantitative about that and say, okay, how exactly do core residues constrain the evolution of sites? And is it really the fact that these residues are in the core, or is it maybe the fact that they have maybe many contacts, or maybe is it the fact that they don't have as much flexibility as surface residues? And this whole area was kicked off, from my perspective, by a paper. Oh, sorry. Okay. Let me go to that paper in a second. I forgot the next slide that was coming. So how we will measure structure in this talk is we will look at 2 quantities primarily. One is the relative solvent accessibility. So that just measures how exposed a particular residue is to solvent. So if you look at this residue here, for example, it is on the surface, and these are water molecules, and most of the residue can be touched by water molecules. So then this has high solvent accessibility, and relative solvent accessibility just means that we normalize this to 1. So if the side chain is completely exposed to solvent, then the relative solvent accessibility would be 1. And down here, this residue in the interior cannot be touched by solvent at all, so its relative solvent accessibility is 0. And then we also look at a different quantity, which is the weighted contact number, or contact numbers more generally. And contact numbers just count the number of contacts that a residue has. So this in the interior has a high contact number, in the exterior has a low contact number, and the weighted contact number in particular weights all possible contacts in the protein with a 1 over distance squared relationship. So the residues far away to this residue, they will still contribute to the contact number, but it will be dampened by 1 over distance squared. And then these ones around here will contribute quite highly. And contact number and solvent accessibility are closely related, but they're also somewhat different. And so much of the recent discussion has been focused on these two quantities and how similar or different they are. And so now we're getting to where I was a minute ago. So this whole topic has been kicked off by this paper by Franzosa and Jar from 2009, which said that evolutionary rate increases linearly with solvent accessibility. So in their paper, their measure of evolutionary rate was dn over ds, so the rate of non-synonymous to synonymous substitutions. As I said, there's many different ways that we can measure site-specific evolutionary rates. I'm mostly glossing over it. In all cases, it will just measure how quickly residues change at individual sites. So what they did, Franzosa and Jar, is they took all the genes in yeast that they could map onto a structure, and then they binned all sites in the yeast genome into bins of different relative solvent accessibilities. And you see here in the gray in background the number of residues in each relative solvent accessibility bin. So there's a lot of core residues and not that many surface residues. And then they found that if they calculated dn over ds in each bin, they found this beautiful linear relationship, that the further you go out on the protein structure, the more variable sites are, and it seems to be mostly linear. Okay, and they also tested contact number in their analysis, and they wrote this sentence. They said that it's solvent accessibility, not contact number, that determines primarily the variability of a site. So I quote them here: the most significant correlation belongs to RSA, R equals 0.126. Correlations with certain packaging-related properties are close behind in strength. For example, the contact number at R equals minus 0.118. So I want to know two things here. First of all, contact number correlations always go the other way, right? Because a high contact number means low solvent accessibility. So if you have a positive correlation with solvent accessibility, then you have a negative correlation with contact number. And the other thing that I would like to highlight is that these correlations are actually quite low, right? 0.126, 0.118. That's really a pretty bad correlation. And as you'll see in a second with different data sets, we can actually do much better. So there comes the next paper from 2014, which argued for local packing rather than contact number. So this was Ye et al. MBE 2014, and that Julian Echave was the senior author on this paper. So they wrote, not only the average unique contributions of both local packing density, LPD, measures are larger than that of RSA, but if one were to choose one variable to model structural constraints, LPD measures are a safer bet with an odds ratio of approximately 3:1. So in their paper, which used a completely different data set, and I'll show you the data in a few slides, and a different measure of evolutionary rate, they found that local packing density outcompeted RSA.
Sorry, is LPD WCN?
So yes, I forgot to mention this here. So they, as the measure they used in this particular paper, they used contact number and weighted contact number. Yeah, and they called both measures of local packing density. And in 2 slides, we'll get to the question whether that is actually justified. But yes, for now, that's what they use, WCN as local packing density.
And while I have you interrupted, I mean, the R sure looked a whole lot better than sort of 1 point something or 0.1 something in the slide you showed. Am I missing something? I mean, it seemed like almost perfect correlation.
In the protein that I showed earlier?
In the plot you showed.
Oh, but remember that was binned, right? So one of my pet peeves, we need to be careful when we bin. So each bin is an average over many sites, right? And then the average falls beautifully onto a line. But if you calculate a correlation, you're not calculating a correlation on these points. You're calculating a correlation on the underlying raw data, and that's much more noisy.
Well, if I was one of those people who just looks at the figures and not at the text, I would be a lot more impressed. Okay, got it. Thank you.
Okay, so this was Ye. They did local packing. Now, you can ask, okay, RSA, local packing, maybe there's other things, right? First of all, local packing or contact number and RSA, those are all really crude measures that don't take advantage at all of all the sophisticated knowledge that we have about protein biochemistry. Maybe we could stick the entire protein structure at an atomistic level into a protein design software and try to systematically calculate how tolerant sites are to substitutions. That could be one way of getting better results. And maybe also, so both RSA and contact number describe the static structure, but maybe what matters is how different regions in the protein move. And so maybe we have to look at the dynamics and see which sites are variable and which sites are constrained. And I could imagine that a more variable site would be more easily mutable. And so this is a question that my lab looked at around the same time that the Ye et al. Paper came out. So that was Shamoradi et al. And we looked at all sorts of different measures besides RSA and WCN. So we also looked at flexibility measures, and we tried protein design. And we found that RSA and WCN performed the best. So this is 9 protein structures that we looked at. And so here we're showing the correlation with, so we used entropy as a variable, as a measure of site variability here rather than evolutionary rate, but the results are roughly the same for evolutionary rate. And so we looked at RSA, WCN, then the variability in the side chain angle chi1. So that tells us how much the side chains move around. Root mean square fluctuations. If you stick the protein in a molecular dynamic simulations and you ask how much does an individual residue fluctuate around. We looked at the B factor and also what we call designed entropy, where we designed protein structures and just asked how variable are individual sites in designed alignments. And you see overall there's a declining trend, so the correlations get weaker as we go further over here. So the flexibility and the design didn't quite work as well as the RSA and the WCN. And there's a paper by Julian Echave, a theoretical paper that also gives a theoretical argument why maybe flexibility is not the right measure and packing or WCN might be the better measure to predict site variability. However, so this paper argued basically again that RSA and WCN were maybe roughly the same and not so different after all. And then there was another paper by Echave where he said, well, no, WCN really wins if you calculate the side chain contact density. So you don't look, most people look at the C alpha to calculate the distance between residues, but if we calculate it based on the center of mass of the side chain, or I think the geometric center of the side chain, then we get the best performance. So they said, we use the mechanistic stress model. So that's this theoretical model that predicts that site-specific rates of evolution depend on contact density, but they use this now to predict that site-specific rates of evolution depend on the side chain contact density. And they also say side chain WCN outperforms C alpha WCN and RSA, and the independent contributions of the latter are negligible. Okay. And then the last paper in this sequence is a paper from my lab again, where we said, okay, that's true, but it is complicated because actually the commonly used measures of packing density, which is mostly WCN, capture non-local effects of amino acid packing in a protein. So you may recall that the WCN is defined as a weighted average over the distances to all residues in the protein. So it's inherently a non-local measure. And so in this paper, we showed that there's actually a local and a non-local component. And the local component looks very similar to RSA, actually, or to other more local measures of packing. And then there's this additional non-local component that measures the overall distribution of amino acids in the entire protein. And combined, that measure performs, it's really the best measure we have to predict how variable a site is going to be, but we don't quite yet understand why it does so and what exactly it does. In any case, I'll now show you our best understanding of how those two measures, WCN and RSA, predict evolutionary rates. And the main thing that we found is that all structural predictors perform roughly similarly for a given protein, right? So what I'm showing you here is the absolute rate WCN correlation. So each dot is now a protein, and I'm showing you the correlation coefficient between rate and WCN along the x-axis, and absolute just so we get rid of this sign problem that WCN correlations are negative and RSA correlations are positive. And here I show you that the rate RSA correlation, and you do see that overall the dots fall below the 1-1 line. So WCN on average performs better than RSA. However, there are a few proteins that fall on the other side. And most importantly, there's a very strong correlation between these two correlations, right? So some proteins, and I've highlighted a few. So this one here, 1R44, its correlation is almost 0.75 along this axis, and it's still about 0.7 along this axis, right? So if WCN works, then RSA works. And I'm showing you here the structure, and mapped onto that structure how variable or conserved I would expect sites to be based on where they are in the structure. And you see mostly the colors are white, which means very few sites are more variable than expected or more conserved than expected. Then here I have one in the middle. That's the E. coli exonucleose that we looked at earlier. And you see there's some more red and some more blue. And you may recall that the active site was around here. So you can see actually there's additional conservation around the active site that we wouldn't have expected just based on the structure. And then there's one down here. And there's a lot of blue and a lot of red. And one thing that you may notice is this surface loop here entirely in blue. And so here the active site is right here. So at least this surface loop is probably highly constrained because the active site is here. Okay, and so this brings us to the take-home message of this part and leads us to the next part. So the take-home message of this part is that packing and solvent accessibility are good predictors of rate in some proteins, but not in others. And for sure, one of the reasons why it may not be as good of a predictor is if the active site in a protein imposes strong additional constraints. Now, I don't want to give the impression that it's only active sites that cause this difference between very conserved and very variable, but certainly active sites play some role in this spread. And so that's the next part. How does protein function constrain site-specific evolution? And specifically, we're looking at active sites. And so the work that I'm presenting was mostly done by my current graduate student, Ben Jack. And this entire project was actually in collaboration with Julian Echave, who is a co-author on the paper.
So before we move on, can you say a little bit more about this non-local component? I mean, it seems a little mysterious to me. I mean, like I can understand the things that are very close by, but you have sort of some interpretation of what the non-local, I mean, you said something about like the aggregate collection of amino acids that are in the neighborhood, but I don't, biochemically, I guess I don't really see how that would work if they're not touching.
So it's the definition of the contact number, right? If I go back to
I see that you have this, you have this inverse D, and there is, like, I see that, somewhat far away amino acids can matter.
So in fact, this by using this square, you actually, if you think in terms of spheres around the focal residue, right? Each sphere in total makes exactly the same contribution because the surface area of the sphere increases with the square of the distance, and we're weighting all the residues in the sphere with the inverse square, right? So we did things in this paper like we said, okay, what if we use a different exponent? So if you have a higher exponent, say 3, 4, 5, then you're really putting more emphasis on the local packing and less emphasis on the residues further away, right? Versus if you have a lower exponent here, then you'd actually put more emphasis on the residues further away and less emphasis on the local residues. And it turns out that the 2 is very close to the optimum exponent here that maximizes the correlation. So if I make my WCN maximally non-local, then I get the best correlation with the evolutionary rates.
I guess I'm just surprised that the things that aren't directly touching have some, that they matter in some way that can be productively modeled in this sense. I mean, I understand that other related residues like can change the local geometry, but that seems sort of that it wouldn't be a productive thing to model with just
well, so one thing is that the WCN is actually a great measure of how central you are in the protein. And the most central part often is close to the active site, which often is the most conserved site. And so WCN may capture some of that. And that actually is directly related to the next part of the talk. So maybe I can just continue and then we can maybe loop back to that. Does that sound reasonable?
Yeah, that sounds great. I'm just trying to do a good job of interrupting you as much as possible.
And I'm totally okay with that. It helps me figure out what to talk about. Okay, so next part. So let's go back to this particular structure that we had looked at earlier, right? And we have the catalytic residue, and now we want to somehow measure how that, actually it's more than one, it's multiple catalytic residues that are all in this pocket, in this structure. How do they affect evolutionary variation in the structure? Well, a very simple measure that we can look at is the distance, right? We just, for each residue in the structure, say this one, we calculate its distance to the nearest catalytic residue, whichever it may be, right? And so then there's some structures that are quite near, some residues that are nearby and some that are further away. And you can immediately see that if there's a longer line, the line points typically to a lighter color, right? So there seems to be an effect of distance on evolutionary variation. And we did this analysis on 524 enzyme structures. And if we do that, and if we throw all the data together, then we get this result where the relative rate, so that's the rate at which a site varies relative to all the sites in the given structure, increases almost linearly with the distance to the catalytic residue up to about 30, 32 angstroms. So you see here the rate is almost zero right at the catalytic residue, right? Catalytic residues are incredibly conserved relative to a typical site in a protein. And then the further you go away, the more variable they are. And then it looks here like this really sharp bend. But actually what is happening here is we're just running out of data, because if you look at where the residues are as a function of distance, 80% of the residues in this data set are actually captured up to this point. So almost let's say the vast majority of all residues in this enzyme data set seem to experience some amount of constraint from the presence of an active site somewhat nearby, but not actually that nearby. And so then what you see here, these dashed lines, they indicate shells. So we can think of shells around the catalytic residue, right? At distance 0, we add a catalytic residue, and then you see the density here goes to almost 0. That's because right here we've gone exactly a distance out so that we've left the catalytic residue, but we have not yet reached the first neighboring residue. And then the first neighboring residue is in this distance range, and you see again there's a bit of a dip right towards the end. And then we have the second neighbors, the third neighbors, the fourth neighbors, and so on, right? And so basically what this says, so we have 1, 2, 3, 4, 5. Up to this point, this is the fifth neighbor of a catalytic residue, and the fifth neighbor still seems to feel just a tiny bit of additional conservation from the fact that there is an active site over here. Okay, and if we want to model the empirical rate, it turns out we need the distance in addition to structure to get a good prediction. So what I'm showing you here is the average relative rate, right? So this is the average of the rate in each shell, and this is the measured result. And as I showed you in the first part of the talk, if we just use RSA and WCN, we can build a pretty good model, and that's true. But these models systematically fail very close to the active site, right? So the purely structural model would predict that a catalytic residue is conserved with about at a value of 0.5, right? Evolves half as fast as the average site in a protein, but the actual catalytic residue is much, much more conserved. And then if we just throw in this one additional predictor, the distance to the nearest catalytic residue, then we get almost perfect predictions on average. So remember again the binning issue. So this is the average in each bin, right? So this doesn't mean that we now can perfectly predict the evolutionary rate at each site. Our overall variance explained is somewhere around 44%, if I remember correctly. But this shows that there's basically no bias anymore, right? So this is a fairly biased estimator near active sites, the purely structural model, and the structure plus distance model is a virtually unbiased estimator. And note, even like far away, it still has this corrective effect, where far away from the active site, WCN and RSA actually underestimate the rate, and once we throw distance in, it gets it right.
Sorry, can you just say what is the form of this estimator? Is it a regression sort of
It's just a linear model, yeah. So we just say we regress rate against WCN and RSA, or rate against WCN and RSA and distance. And we can do this. So in the paper, so this paper is about to come out in maybe 2 or 3 weeks. We did this systematically both on the entire data set thrown together at one and on each individual protein structure, and the results generally hold up. I mean, there is again this effect that correlations are higher in some structures and not as high in others. But on average, we explain something like 44% of the variance. Did that answer the question?
Perfectly.
Okay. So then let's just look at a few examples where, so I've mentioned earlier that when the active site is in the core of the protein, then we don't really know whether sites are conserved because they're in the core of the protein or sites are conserved because they're near the active site. So one way to disentangle that is to specifically look at proteins where the active site is on the outside. And I'm just highlighting 2 examples here. The one is PDB structure 1D06, and I'm first showing you the various correlations of the different quantities. So K stands for evolutionary rate. So RSA and WCN are highly correlated, as you would expect. RSA and WCN are very weakly correlated with distance, right? So we've picked a structure where WCN and RSA do not correlate with distance because the active site is on the surface. And then you see that rate correlates quite strongly with distance in this protein and correlates more weakly with WCN and RSA. And so this is what it looks like, the relative rate as a function of the distance to the catalytic residue, and it seems to just linearly go up. And if we map it onto the structure, you see, so here now blue means conserved and yellow means variable. And very clearly the active site is quite conserved and the variable regions in this protein are on the other side of the structure. I can show you this blown up and you see how beautifully it just goes more and more variable as we move away from the active site. Okay, a second example, 1-SMN. So again, RSA and WCN are very highly correlated. Distance is basically uncorrelated to WCN and somewhat correlated to RSA. And then WCN, RSA, and distance all have comparable explanatory power. But again, if you plot the rate as a function of the distance, actually in this particular case, it just seems perfectly linear. It's quite a surprising result. And this is what it looks like in the structure. And again, blown up. So the active site is over here. And clearly it's the most conserved region of the protein. And the most variable region, this may be somewhat of a surprise, is actually this interface with the protein is a dimer, so this interface with itself, right? This is not really a solvent-exposed region because the protein needs to bind to itself here, but this is the most variable part of the structure. Okay, and so this prompted us to also look at interface residues versus catalytic residues. So our data set of 524 enzymes has both catalytic residues and has protein-protein interfaces. And we found, so let's look at the left here first. All sites on average have a rate of about 0.1. Catalytic residues are extremely conserved, right? The rate is, sorry, all have a rate of about 1. And then catalytic residues have a rate of about 0.1. So they're 10 times more conserved than your average site. But interfaces only have an average rate of 0.9, right? So the rate of evolution of an interface residue is actually not that different from any random site that you may pick out. And so we tested this particular case in a second data set where it's non-enzyme. So we just took protein-protein complexes. So we cannot make any statement about catalytic residues, but we can make a statement about interface versus non-interface residues. And the distance here is a bit bigger, but again, the interface residues are much more variable than are the catalytic residues in enzymes. And we also looked at the variance explained. So catalytic residues on their own, and this is, if I recall correctly, it's a delta R squared. So this is controlling for structure. So this is how much of an improvement do I get in my model if I have already a model that contains WCN and RSA, and now I throw distance on top. So catalytic residues add about 5% of variance explained on top of the purely structural models, versus interface residues add only between 1 and maybe 2.5 or 3, this maybe 3% of variance explained in addition. So this is a distance model where we calculate distance to interface, right? So clearly there is some additional constraint in interfaces, but it's much weaker than what we see in the catalytic residues. Okay, so this concludes part 2. And there the take-home message is that catalytic residues generate long-range selection gradients covering most of a typical enzyme structure. And now I'm going to part 3, and part 3 continues right where we left off and says, okay, let's Can we learn more about protein-protein interfaces? And so the question here is, how do protein-protein interfaces evolve? And that entire work was in collaboration with the Marcotte lab, and I'll show you in a second what they did. They did great experimental work that I cannot claim any credit for, but I'm going to present it anyway because it's great. And then we did protein evolution simulations, and that was a former grad student of mine, Austin Meyer, who did that. Okay, but before we can get to the protein-protein interface question, let's take a detour to what the Marcotte lab did. And it may initially look like it's a completely different topic, but I promise we'll go back to proteins. Okay, so the Marcotte lab asked, can human genes function in yeast? And so what they did is they systematically replaced essential genes in yeast with the human ortholog. So they looked at all essential genes in yeast, found the human one-to-one orthologs. There's an overlap of 547 genes that satisfy both criteria. And then 469 of them, they actually systematically, one by one, stuck into the yeast and then assayed whether the yeast was still viable with this human gene stuck in place of the yeast gene. And what they found is that in about half of the cases, the yeast gene is replaceable, right? About 200 of the genes were replaceable, and about actually exactly 224 were not replaceable. And then there's this toxic category that I'll gloss over, but you can read it up in the paper if you want to know details about that. But the main take-home message from this experiment was that about half of the genes are replaceable. And so now if you come across this finding as a good evolutionary biologist, you would say, oh, it has to be sequence divergence, right? Surely the genes that have not diverged so much between humans and yeast, they're going to be replaceable, and the ones that have diverged a lot, they're probably not going to be replaceable. And in the extremes, that is correct, but it's also not quite as simple. So this plot, let's look at the red line first. So this is the fraction replacing. So it's the axis on the right here from 0 to 1, right? Fraction of genes replacing as a function of the amino acid identity. And if you're all the way over here between 0 and 10, so completely diverged, then still about 30% of the genes replace. That's actually quite remarkable. But then over here, if the gene hasn't diverged, then 100% replace. That's maybe what you would expect. But also, there's very few genes in that category. So this is the axis on the left. The count is the number of genes that fall into these amino acid identity bins. And as you can see, the vast majority of genes fall between 20 and 50% amino acid identity. And there, the replaceability is about 50%, right? So if you take a random gene from the yeast genome and you replace it, a random essential gene, you replace it with the corresponding human ortholog, then you have a 50% chance that it will or will not replace. And so sequence identity is not that predictive. So the Marcotte lab also did a systematic bioinformatics analysis trying to find which types of quantities would predict replaceability. So the next slide is going to be a bit of a monster, but so this is all the different predictors that they tried, right? Many different predictors, but they're color-coded here. So they're sorted from really good to really bad. Over here, the gray one over here, that's actually a combined predictor. So this is overall, they got an area under the curve in the ROC curve of about 83%. So overall, they could predict quite well whether a given gene was replaceable or not. But you see, if you just look at the color coding, most of the color is orange, so that's network. So the best predictors of whether genes were replaceable had something to do with the network structure. And one way that we can summarize this is that genes tend to be replaceable in modules. And I'm going to show this to you visually. So in the following few slides, I'll use the color coding where blue means not replaceable, yellow means replaceable, and then white means not tested. For example, in this trick chaperonin, all the genes that they tested were not replaceable. Or in this genome replication origin complex, all the genes that they tested were not replaceable. Now, in this sterile biosynthesis pathway, almost every gene that they tested was replaceable, with the exception of this group down here that was not replaceable. This maybe you might say, okay, these are all enzymes that do some biological function, versus the previous things were complexes, so maybe it's just complex versus enzyme. But that's not correct, because there's other complexes, like for example the proteasome, where almost every protein in this structure is replaceable. Right, the proteasome has this structure with a lid and a base and a core. So the core is this ring of proteins, and then the base is this additional ring of proteins, and then you have the lid. And with the exception of the core, where most proteins are not replaceable, and oh, by the way, it has a symmetric structure. So from here on up and from here on down, it's just a mirror image of itself. And you see the proteins in this ring are not really replaceable, but then everything else mostly is replaceable. And so this is a complex molecular machine that has diverged almost 2 billion years between yeast and humans, and yet you can just take human structures, human proteins, one by one and stick them into yeast, and the yeast will be just fine. And so it was findings like these that motivated us to ask, okay, what does that tell us about protein-protein interfaces, right? If we have a molecular machine and it diverges over time under the constraint that it needs to continue binding, how does the interface diverge, and does it continue to bind to the ancestral state? And so there we came up with this hypothesis that at least in cases where we have protein-protein interactions, if they need to be conserved, then this may cause replaceability. And so this is where my lab comes into the picture, where we now set up a simulation to test this idea. And so this is how we set up the simulation. We take a pair of proteins, A and B, that need to bind to each other, and then we simulate evolution. So we have a computational model of the structural divergence of this protein interface. And so the proteins diverge and the interface diverges, right? So we impose that A and B remain stable throughout the simulation, and also that A and B continue binding throughout the simulation. And then we ask, okay, if we take the evolved A, does it bind to the ancestral B? And likewise, if we take the ancestral A, does it bind to the evolved B? So that's our treatment simulation. And then the control simulation evolves A and B without binding, right? So we pull A and B apart, so we still impose the constraint that A and B need to be stable as they evolve, but then we just let them evolve individually. And then we ask, does the evolved A bind to the ancestral B, or does the evolved B bind to the ancestral A? And this is our result. So if we run many replicates, and then we ask, what's the likelihood that the evolved protein binds to its ancestor? So 0 means the likelihood is 0, it doesn't bind, none of the replicates bind anymore. 100 means they all bind. And we plot this as divergence, right? So over here at 0, that's where we're starting out. So we have 100% chance that the evolved protein binds to its ancestor because it hasn't changed. And then over here, we go out to about 80% amino acid divergence, so 20% identity. And this is the non-bound case. This is the control case, right? And as you can see, as divergence accumulates, the percentage of structures that still bind declines very nicely, exactly as one probably would have expected. However, now if we impose the condition that the proteins continue binding, then it looks very different. And we have two different scenarios here. One is wild type and one is low stability. Low stability means we are actually not imposing a stability constraint on the protein. We let it evolve however it wants, but we do continue imposing a binding constraint. And those two curves in these particular simulations look quite similar. But the important point is that we can easily go out to an amino acid identity of below 50 or below 40% and have lost very little binding. In case anybody wonders, these black dots here, so this is a survival curve, right? So that means right census data. The way we do this is we simulate the evolution for a set number of generations. I believe we did 1,000 generations in this case. And then at the very end, we have some divergence, and that's the divergence that we get. And so, for example, in this particular case here, it ended at a divergence of about 75%, and it was still binding at that point. So we cannot really make any statement about what happened past that point. We just know it was still binding at that point. So this is all these dots here mean for those particular simulations, we could not go any further. At that identity, the structures were still binding to the ancestral state, and that was when we ended the simulation. Okay, so now you might ask, well, so does this happen because the protein accumulates a lot of mutations, but they're not in the interface? Maybe they're just in the rest of the protein. And so we tested that by making a similar plot, but separately for interface and non-interface residues. And so on the left, we see divergence in interface sites. On the right, we see divergence in non-interface sites. And so we find that even with substantial divergence in the interface sites, the evolved proteins bind their ancestors. There's almost no difference between this graph and this graph. Well, I mean, it declines a little further. So clearly divergence in the interface sites does push you a little quicker away from binding to the ancestor, but we can accumulate 40 to 50% divergence in the interface and still bind to the ancestral state. Now, importantly, we count divergence here as just has the amino acid changed or not, right? So if you change a leucine to an isoleucine, then we would count that as a change, but biochemically, not that much has happened. And so the interpretation, therefore, is that we think that what happens with conserved interfaces, so if selection requires the two protein partners to continue binding, then the biochemical signature of the interface somehow remains conserved, even if the amino acid identity changes. And that's why the sequence can diverge, but the protein still can bind to its ancestral state. And so that's the take-home message from this part. As protein-protein interfaces diverge, they seem to maintain biochemical similarity to their ancestral state. Okay, and I'm right on time, and that was my last slide. So let me just thank all the people. So I've listed everybody currently in the lab, and I've underlined the people that did the work. So Austin Meyer and Amir Shamoradi, who used to be graduate students, and Austin Meyer is now at Texas Tech, and Amir Shamoradi is still at UT, but is a postdoc in a different unit at UT now. They did a lot. So Amir did most of the work in the first part, and Austin was involved in all these projects and did all the work in the third part. And then my current postdoc, Ashley Teufel, she's continued on where Austin has left off. And so she's continuing these kinds of simulations and investigating the divergence of protein-protein interfaces in more detail. And then Ben Jack did all the work in the second part with the effect of catalytic residues on site variability. And then collaborators already pointed out Julian Echave and Ed Marcotte from the Marcotte lab. who did all the yeast work, and the people in the Marcotte lab who did all the yeast work were Ashik Kachru and John Lauren. And I misspelled his name. I think he doesn't have an H there. And then funding from NSF, NIH, DITRA, and the Army Research Office. Thanks a lot.
All right, that was a fantastic and very clear talk, Claus. Thank you for that.
Thank you.
So, I mean, regarding the first two parts of the talk, I mean, and maybe the third a little bit as well. I mean, you've done a phenomenal job of nailing down ways of predicting amino acid mutability, but there's sort of another dimension, which is, what are things mutating to? And I guess you alluded to it a little bit in the tail end of the talk, but do you see any way of extending this to talking about amino acid properties of substitution?
So I'm not sure I understand the question. Are you asking, can we predict what kind of mutations might happen at individual sites, or what exactly is the question, I guess?
Yeah. So I mean, I guess I think of substitution as being like there's sort of a rate component, but there's also what is the matrix. So
yeah.
If the per site, clearly from what you're talking about, one could develop a per site model that would have a rate that's determined by what the surrounding amino acids are in the context of the structural alignment. But could we also have a substitution matrix?
So I personally am actually quite skeptical. I'm aware that this is widely used in phylogenetic inference, that there's basically a research program that says we try to come up with the best substitution matrix. I'm very skeptical about that because I think that most substitution matrices that are not site-specific are just way too general. And then the ones that are site-specific are basically only hypothetical. You cannot really know what the substitution matrix would be unless you had well, in theory, maybe we could calculate it from an all-atom model of protein structure, but we cannot currently do that. So, yeah, this is almost a separate talk, but since you brought it up, there was just recently, actually, Richard Goldstein is almost certainly going to talk about that, because I was just about to pull up his latest paper. He just published a paper in Protein Science, I believe, that addresses that point to some extent. So maybe just wait for his talk. But let me just say one empirical thing that we have found. If we look at amino acid, if we look at protein sequence alignments, we find that typically there is 3 to 6 amino acids that tend to be allowed at any given site, and they are typically exponentially distributed. So the most abundant one, well, the second most abundant one is exponentially less abundant than the first one, and then the third one is exponentially less abundant one than the second one, and so on. So you have these very rapidly declining preference patterns at each site, and that is not at all like what these amino acid substitution matrices look like, that basically average over all sites. And there's an interesting twist that leads back to maybe the topic. One paper that we wrote in 2011 that I didn't go into, but actually that one I could pull up quickly if we have a few minutes and I find my web browser. Okay. 2011, it's already quite old. There. So the question is, so this was one of the first paper. So this is Ramsey et al., Genetics 2011, the relationship between relative solvent accessibility and evolutionary rate in protein evolution. And so that was shortly after we we had seen the Franzosa and Jar paper, and we said, okay, if surface sites are more variable and core sites are more conserved, can we take a model of amino acid propensities at surface or core sites and use that to predict rates? And it turns out that didn't work at all. And so that is going back to the question that you had, if the figures come up here. So basically, if you look at all surface residues, okay, so this is the pattern, right? The rate increases linearly with RSA. And this is the pattern. Let's just look at B. If I take a large number of proteins and I bin residues by RSA, and I look at the overall distribution of amino acids as a function of RSA, and then I use that to predict rate, I find that the surface, sorry, this is kind of in the way here, but so this is the surface, this is the core. I find that the surface is predicted to be the most conserved. The real core is also predicted to be conserved, and this intermediate region is predicted to be the most variable. And there's this theoretical result from Porto et al., part number A, which kind of recapitulates that. It also says that sites in the middle should be the most variable, and then the surface and the core sites should be the most conserved. So that's completely at odds with the observation that actually the surface sites are the most variable, right? And so that is directly caused by averaging over many sites. So if you first average over sites and then predict a rate from the distribution of amino acids, then you get quite conserved of a rate, because right on the surface, there's only a small number of amino acids that will always be used at all surface sites. Now in the core, or like in particular in this intermediate region of an RSA of 0.1 or 0.2, almost any amino acid will, in some protein structure, in some site, be visible in this range. And so if you average over enough sites, it looks like the amino acid distribution is the broadest here, but actually at each given site, it's the most conserved. Does that make sense?
Yeah.
And so this is if we do it right. So if let's just look at A. So this is the relative frequency. Again, we don't see what it says at the bottom, but this is the most abundant amino acid, the second most abundant, the third, the fourth, the fifth. So what we did now was we said, okay, at each site, let's ignore what the amino acid is. Let's just say what is the most abundant, what is the second most abundant, what is the third most abundant, and look at what these distributions look like. And then we do find indeed that they decline the fastest for buried sites, and they decline the slowest for more exposed sites, right? And so here you see this exponential decline. So it's a log scale, and once we're 5 amino acids out, we're already at a frequency of 1%. So buried sites rarely have more than 4 or 5 amino acids that they actually tolerate, and then exposed sites may tolerate some more. This, if you propagate this out, maybe the most exposed sites might allow for 10 or 15 different amino acids. But most importantly, on the surface, it's going to be always the same amino acids, versus in particular intermediate ranges, we have no idea which amino acids it's going to be, but we know that at each individual site, it's going to be only a small number. And so the work that Richard Goldstein and David Pollock are doing right now, it's actually great work towards explaining these kinds of patterns.
Got it. No, that is very interesting, and like you say, rather different than the common assumptions. So there's a question from Ya Yang, and the question is all the research done in 1-to-1 orthologs? And a follow-up question is, any thoughts on protein families with more complicated histories?
So I'm not sure about which project the question is.
My suspicion is that it is the last one, but
So in the yeast project, so far they have done 1-to-1 orthologs. So that's the published part, and they're currently working on many-to-1 orthologs. But I don't really want to speak to that because it's really their project, and also the paper hasn't been written yet. So all I can say is that's something that they're working on, and there will be a paper on many-to-1 eventually. But I can't really speak to any of those results at this point.
It's such a sort of surprisingly simple and fascinating, I mean, simple once you've thought of it, like experiment to try. Though
well, but to be honest, this is a massive experiment, right?
Yeah.
And Ashik worked on this for 3 years. I mean, you need a really dedicated person to stick 547 genes into yeast and then assay them all. And I mean, it's not just 547 assays, right? Like you have to verify that it worked correctly. And then, so if since we're talking about this, so the toxic ones are the ones where the initial expression level is off. But then if you modify the expression level, it turns out that they actually can rescue the yeast and so on. I mean, this is just a really massive experiment.
Yeah, I guess I meant simple in conception, not
yeah.
Sure. Yeah. So I might ask, do you think about what this might look like if one was to try to incorporate these observations that you have in phylogenetic inference? I mean, you said that you didn't believe in aggregate protein substitution matrices across sites, and that's something I also agree with. But do you think that there's any possibility that we will be able to leverage this work for doing inference, or would it matter if we did?
Okay, so the first question, is it possible? I'm sure that it will be possible eventually. I'm just not sure yet how. So I mean, there were some attempts, I think by Rodriguez in particular, to build these sophisticated energy models into phylogenetic inference, and it sort of works, but it takes a huge amount of computational power, and so nobody has really pushed it to a point where it's feasible. And so that comes to the second question. Will that effort ever be worth it? And so far, I guess it hasn't. My suspicion is, as always in science, there's probably at some point going to be somebody who has a clever idea and comes up with a way of making it worthwhile. But right now, I personally haven't seen that yet. But that doesn't mean much. It just means I'm probably not the person who's going to figure it out.
Well, I mean, I don't know. I mean, to play devil's advocate, I mean, if you did have the structure and you could calculate these distances quickly, I mean, doing a phylogenetic analysis with per site rates is not crazy. I mean, if you had a big enough database of the different genes that you would be interested in using and their structures. But
yeah, and I mean, you could use something like RSA or WCN or so to inform yourself on whether you expect a given site to be variable or conserved. I just don't know enough about phylogenetic inference to know whether that would improve your inference.
Yeah. Yeah. Okay, well, I don't see any of the questions. That was a great talk, and I've kept you for a little bit too long, but thank you very much. And you also gave a great little advertisement for Richard Goldstein's talk coming up. I guess it's May 25th or so. So thank you very much, Claus, and see you next time.
Yeah, thanks again for the invitation, and also thanks everybody who listened or watched this. Bye.
Bye.